Hello, my name is Marah Inocencio, and I'm a sophomore undergraduate student studying toxicology in the biochemistry department. This past summer, I had the opportunity to work in Dr. Diana Roopchand's research lab with supervision from my PhD candidate, Kevin Tveder. The project I worked on involved studying how dietary polyphenols found in grapes may be beneficial to combat chronic diseases like type 2 diabetes and metabolic syndrome. Metabolic syndrome, or MET-S, and type 2 diabetes are characterized by insulin resistance, high blood glucose levels, high serum triglyceride levels, high blood pressure, and overweight or obesity. Type 2 diabetes and MET-S are global health concerns that are increasing in prevalence, indicating that existing drug therapies and treatments are inefficient. The research project I'm involved with used a high-fat diet-induced mouse model of metabolic syndrome. We fed 3 different animal mouse strains a high-fat diet or a high-fat diet supplemented with 0.5% grape polyphenols by weight for 12 weeks. Therefore, we had 6 groups in total. Dr. Roopchand's lab had previously shown that grape polyphenols, compounds found in plant products with antioxidant properties, cause metabolic improvements in models of metabolic syndrome and type 2 diabetes. Previous studies showed that these poorly absorbed grape polyphenols alter the gut microbial community and that these gut microbiome changes were associated with phenotypic improvements like enhanced glucose metabolism. The lab identified a second mechanism that suggests grape polyphenols alter the gut microbiome and bile acid profile to suppress intestinal signaling of bile acids to bile acid receptor Farnesoid X-receptor, or FXR. Suppression of intestinal FXR is also associated with improved glucose clearance with lowering gene expression of ceramide synthesis genes in intestine and liver tissues. Before I describe the data, I want to provide a brief background regarding the importance of bile acids and bile acid signaling. Bile acids are metabolites produced from cholesterol in the liver, which are secreted from the gallbladder as primary bile acids into the intestine to digest dietary fats and lipophilic vitamins. However, post-digestion, bile acids play a pivotal role in modulating bile acid production and glucose metabolism via the enterohepatic circulation and bile acid signaling to FXR. In the gut, primary bile acids are converted to secondary bile acids by gut bacteria who possess enzymes to do so. Depending on whether the primary bile acids are converted to secondary bile acids by gut microbes, these bile acid species have differential effects on FXR activity. Activation of FXR in the intestine transcribes genes FGF15, fibroblast growth factor 15, which sends signals to an enzyme in the liver, CYP7A1, that inhibits expression of primary bile acid synthesis. FXR is also involved in transcribing a group of genes needed for ceramide synthesis, including SIRS4, SMPD1, SMPD3, and SBTLC2. Ceramides are a type of sphingolipids that may be a driving force in the development of cardiometabolic diseases when excess ceramides accumulate in tissues and in circulation. Studies suggest that accumulation of ceramides may lead to chronic inflammation and impaired insulin sensitivity. In order to identify changes in ceramide synthesis and the relationship between ceramide synthesis and the bile acid receptor FXR, we used wild-type mice and mice with FXR knocked out in the intestine, referred to as VFXR mice, and liver, referred to as AFXR. Cre-LoxP recombinase technology was utilized to mate mice to drive deletion of FXR from the tissue in each respective knockout mice, liver or intestine. Each strain was then fed with either a high-fat diet or a high-fat diet supplemented with 0.5% GPs to test if GPs can still promote improved metabolic outcomes as we have observed in the control wild-type mice. We analyzed phenotypic traits such as body weight, food intake, and body composition, metabolic tests, and collected fecal samples at various time points, as shown in the diet intervention timeline under experimental design. In order to confirm which linearmates had FXR deleted, genotyping and genomic DNA extraction were performed on ear tissue of 7-week-old mice. PCR would amplify the presence of Cre2, as seen in the gel image with 400 base pairs, indicating that FXR was knocked out. The flowchart under experimental design shows the breeding of a wild-type mouse with a liver-specific or intestine-specific FXR knockout mouse, producing half of the offspring to be knockouts via the enzyme Cre that eliminates the presence of FXR. Quantitative PCR was done to confirm deletion of FXR in the liver in all 6 groups of mice, as shown in the figure showing results of FXR expression. RNA was extracted from liver tissue and was made equimolar to convert to cDNA via reverse transcriptase, which was then ultimately used to perform Q PCR. Using the same method, real-time Q PCR was then performed to study the relative gene expression of ceramide genes in the wild-type groups. According to the data, there was a significant decrease in hepatic expression of ceramide genes SMPD1 and SBTLC2 in the wild-type groups fed a high-fat diet with the Grape Polyphenols. This indicates that Grape Polyphenols may influence the gut microbiome and bile acid profile in a way that downregulates FXR transcriptional activity. Analyzing hepatic gene expression of ceramide genes may suggest lower accumulation of ceramides, especially because the liver is a major organ for ceramide production. However, further research needs to be done to analyze the ceramide profile in the serum and metabolic tissues. Studying ceramide and bile acid profiles in the liver and ileal content are currently ongoing through the performance of targeted LC-MS. Further research with this method may include analyzing bile acids in hepatic and intestinal tissues in all mice groups. Compiling results from the insulin tolerance tests may also be done to draw conclusions in the relationship between the accumulation of ceramides and bodily dysfunctions that may ultimately lead to insulin resistance. Following this, cell culture and organoid work may be performed for a deeper and closer study on bile acid profiles in ileal tissue from the VFXR and wild-type mouse strains. Enlightenment from these future studies may explain the connection between grape polyphenols and their effects on the bile acid and ceramide profiles. This may ultimately bring research closer to concluding the mechanism behind how these poorly absorbed grape polyphenols can decrease risk of cardiometabolic diseases. Given this study and experience, I'd like to offer my thanks and gratitude to my mentor and the advisor of this project, our faculty advisor, and to our graduate student mentors and fellow undergraduate students for assisting me in this research experience. A special thank you is also extended to the funding committees for supporting the growth of this project. I would especially like to express my appreciation for the Douglas Residential College's Project Super program for giving us a platform to present and work in summer research. Thank you.